This is a short video on DNA structure and DNA replication. And I'm going to use the application or gizmos to model how DNA is copied. Okay, here we have a sample strand of DNA. We have the sugar, the pentagonal deoxyribose sugar that's attached to the phosphates. Okay, sugars and phosphates are making up those sides, that backbone of a DNA structure. And then you have the code within the double helix, the DNA. The code is in these nitrogen bases or nitrogenous bases. You have adenine pairing with thymine. You have cytosine pairing with guanine. What happens when DNA replication occurs in interphase, in the S phase of interphase in the cell cycle, you have an enzyme, DNA helicase. And that DNA helicase is going to unzip or unwind the DNA so that DNA polymerase can go ahead and pair up. The complement can get the complement to each of these nitrogen bases to create that new strand of DNA, or the two new strands of DNA. So again, we have that base pairing that we're using. Okay, we're going to take these nucleotides to complete new strands. And so again, adenine pairs with thymine, and we have cytosine pairing with guanine. So T and A, and T and A, and G and C, and G and C. These are we're using that base pairing rule, those complementary base pairs to create new strands of DNA. And the DNA polymerase is a very important enzyme that is going to allow this process to happen. And we had discussed the cell cycle earlier in the semester, and the copying of DNA is happening during interphase, that synthesis phase of interphase. And it's a very important process. So we're going to complete our strand by using that base pairing rule. Okay, you have the enzymes. You have enzymes that are going to unzip the original strand, and then you have an enzyme that is going to help create the two new strands. And if you look at these two new strands, they are identical. You can see that these two new strands of DNA after DNA replication are identical.